Previous year questions, you can see that almost 3 to 4 questions are very common from this chapter. Tunica Vaginalis has two layers. One is the parietal layer and another one is the visceral layer. Visceral layer stays close to the testis and the parietal layer stays out. Hello Bachcha Badi, Namaskar and welcome back to my YouTube channel Sahil Educations. Today we are going to start a fresh new chapter. The chapter's name is Reproduction in Human. Reproduction in Human for your NEET exams. So if you go for the previous year's questions, you can see that almost 3 to 4 questions are very common from this chapter. So you have to understand the concept behind this chapter. You have to understand all the aspects of this chapter. Basically, we are going to start from the male reproductive system today. Male reproductive system. To understand the male reproductive system, you have to see the whole diagram here. It has been drawn already. The first one, this one is the urinary bladder, and urinary bladder is just going to eliminate the urine through the process which is known as maturation, through the urethral meatus, through urethra. The urine comes into the urinary bladder, it stores here, and then it enters into the urethra and it goes out of your body through urethral meatus. So urethral meatus and the urethra is basically present in the penis region, and above all this, the male reproductive system. If you talk about the male reproductive system, then there are various kind of organ in the male reproductive system. One is the primary organ. The primary organ in case of male reproductive system is the testis. And the secondary organ, if you talk about the secondary organ of this system, then you can have the Vas Deferens, epididymis, other glands, and penis. So these are the structures which are going to make the whole reproductive system of a male organ. The testis just descends out of the abdominal cavity and it remains in a sac-like structure. That sac-like structure is known as scrotum. So let's start with the dissection, or let's start with the tissues, with the layers which are going to cover this testis. So first one is the skin which is surrounding the testis from the outer part, from the very outer part, which is you call it as a scrotum. And inside this skin you find a smooth muscle. The involuntary muscle, which is known as dartus muscle. And beneath or inside this dartus muscle you get some membranous structure, membrane-like structure, which is known as external spermatic fascia. Inside this spermatic fascia you again get one smooth muscle which is known as cremasteric muscle, which is a smooth muscle. Again entering from this cremasteric muscle you get tunica vaginalis, two layers of tunica vaginalis. One layer is the outer layer which is known as parietal and the inner layer is known as visceral. Tunica vaginalis has two layers. One is the parietal layer and another one is the visceral layer. Visceral layer stays close to the testis and the parietal layer stays out, or stays far away from the testis. So visceral layer, basically this visceral layer is very much attached with the tunica albuginea. So this tunica vaginalis, these two layers of tunica vaginalis are the serous membranes, which are actually the smooth tissues. And now talking about this tunica albuginea. Tunica albuginea is the innermost part, innermost covering above the testis, which actually embeds into the testis to make these structures. These structures are septa. We are going to discuss about these structures in the later part. See, just I'm going to draw here. if you draw a testis, then I'm just drawing this one is the Tunica Albuginea. So this layer is Tunica Albuginea. Tunica Albuginea is a thick layer which just descends down, making these are the septa. These are the septa. Okay. In between two septa, you find some lobule like structures. So basically this space is known as lobule. This space is known as lobule. As per NCERT, you find 15 to 20 lobules in an average in a single testis. And these lobules are going to contain some tubular structures. These tubular structures are known as the seminiferous tubule. These structures are going to be called as seminiferous tubules. These are the tubular structures. Approximately you can say 2 to 3 tubules are going to present in a single lobule, and if we talk about the number of lobules in a single testis, then you can find that 15 to 20 lobules are present in an average in a single testis. Now talking about this seminiferous tubules are basically the place where sperms are going to produce and sperms are going to be matured. After maturation or after production of the sperms inside this testis, these seminiferous tubules will eliminate or will allow the sperm to go out of this testis, and it will go towards this Vas Deferens. So what is the pathway? What is the way actually this sperms follow from testis to the urethral meatus to come out of the body? So inside this seminiferous tubule, the sperms are going to be produced and matured. These seminiferous tubules of each lobules are going to be connected. Inside the testis. Mind it, inside the testis. So this part is known as Rete Testes. Rete Testes. After Rete Testes, these sperms will go out of the testis, and here these ducts are known as ductules efferent, or you can call it as Vasa Eferentia. Vasa Eferentia. After entering into this Vasa Eferentia, the sperms will go towards the epididymis. So epididymis is basically the place where the sperms store in your body. Mind it, epididymis is the place where sperms actually store in your body. So this part is the epididymis. Epididymis is again divided into 3 parts. One, this is the head of epididymis. Head of epididymis. This part is the body, and this part is the tail, or you can call caudal epididymis as well. Okay, caudal epididymis. So where the path till this, the seminiferous tubule where sperms were produced and matured. After maturation, the sperms came out of the seminiferous tubule and entered into Rete Testes. After Rete Testes, the sperms entered into the Vasa Eferentia. After Vasa Eferentia, the sperms entered into the epididymis. In the head region of the epididymis, basically sperms get stored and then it goes out of the body through this caudal part of this epididymis, and this caudal epididymis will now follow the path towards vas deferens. This is known as vas deferens. Okay, so that was the pathway which sperm actually follow to come out of the body. Now there was a question in the previous year that there was a matching question, there was a sequential question that what is the actual path that your sperms are going to follow, or the anatomy of this testis looks like. First one is the seminiferous tubule, then the sequence actually was like the seminiferous tubule, then Rete Testes, then Vasa Eferentia, then epididymis, and then Vas Deferens, and that was the correct answer. Whatever you may get some kind of question upon the anatomy of this testis. If you understand this anatomy of testis, let me know in the comment section, and we will be going to discussing about the glands in the next video. So thank you very much, see you soon.